We're going to do the notes virtually today together since I cannot be here. We have done some of this already with our electrophoresis virtual lab. That was the purple one that you've been working on. So we need to figure out what is DNA fingerprinting / profiling and how can this technology be used. You're going to be filling in your note sheet as we go through the PowerPoint. So restriction enzymes, what are restriction enzymes? We can cut DNA with special enzymes called restriction enzymes. So the first bullet on your note sheet should be special enzymes used to cut DNA. Feel free to pause and rewind this as many times as you need to fill out your note sheet. So this means we can cut up DNA from different people or different organisms and compare them. Why would we want to do that? Well, this allows us to be able to solve crimes, figure out who the murderer is, or who stole something from someone's home, diagnosing diseases, a paternity test, so to discover who the father is, evolutionary relationships, like figuring out that we are related to chimpanzees by looking at our DNA, and several others. Those 5 bullets need to be written in the blank space right below the first 2 bullets. So, what do the bands on a DNA fingerprint represent? They are fragments of DNA. So the little colored white bands is what they are talking about in the picture. Each white band is a section of DNA that has been separated out. What are we looking for? So, why is each person's DNA in a different pattern? We look for specific sections of DNA that vary a lot from person to person. If we examine enough of them, we can narrow it down and point where no one else in the world has the same DNA pattern. No one has your DNA. You are unique in its own way. So when we pass down our genetics to our offspring, half of your DNA is going to go into the child and the father or mother will go in as well, making your DNA unique to you. Remember, you get 23 chromosomes from mom and 23 chromosomes from dad. Okay. Now, how do you get a DNA fingerprint? So now these are the steps that we are going to talk about through gel electrophoresis. And one step is you obtain DNA samples from blood, hair, and semen, etc. We then cut the DNA with the restriction enzymes that we talked about earlier. And then we load the DNA fragments into wells of gel. Okay, those are your wells. That is a pipette that is going to insert some DNA. Okay. Each end of the gel is then hooked up to a charge. There's negative and positive electrodes. Since the DNA is negatively charged, it's going to be attracted to the positive. Negative and positive, just like when you're using magnets, just how you put in batteries. Okay? So the negative DNA is going to be attracted to the positive end. And the fragments of different size travel at different speeds. That's very, very important. If it is heavier, so a bigger piece of DNA, it's obviously going to be weighed down, meaning that it's not going to go as fast or travel as far across the gel. So the small pieces travel the farthest. Okay, there's your gel box. Okay, a power meter, we're going to turn it up to 100 volts. hooking it up, DNA migration from negative to positive. And our fourth step is after DNA is stained, so once it's done through the gel, we're going to stain it to make it visible. The DNA fragments can then be compared because they separate by size. Which size molecules move the farthest, and we stated the small ones. All right. So a recap to help us clarify, why does DNA move down the gel? And we know this because negative, positive direction of the DNA travel, the wells, is going to travel down because DNA is negatively charged, being attracted to the positive. Large, small. Okay, so that's what we look like. This is what we're going to analyze. Okay, what does a match imply? Okay, it's going to give us enough information to evidence from murder trial. Can we figure out if they are guilty? It shows us matches of DNA pairs, and the more matches, the more likely it is that the DNA, the blood, the semen comes from one of the people that has been tested. Okay? Just some examples of comparing some blood samples and how it works. I'm circling the pairs as we go through. Blood sample from the victim. There are 2 there. Blood sample from victim 2. There are 2 there. Alright. And the blood sample from the suspect. And we figure out that O.J. Simpson is the one that did it. Alright. And Brown. Is he? Alright. So these questions are on the back of your paper. The diagram below shows the results of a test that was done using DNA sample from 3 bears of different species. Each DNA sample was cut into fragments using a specific enzyme and placed in the wells as indicated below. The DNA fragments were then separated using gel electrophoresis. According to the chart, which 2 bears are most closely related? So I'm going to pause for a second, you pause the video, and figure out which bear is most likely related. Okay, so I'm going to go with bear 1 and bear 3. Why? Because they have the most pairs that are alike. Bear 1 only has 1 pair of 1 piece of DNA that is different than bear 3. Alright, number 2. Electrophoresis is a method of. Again, take a second. Look at your choices. A, separating DNA fragments. B, indicating the presence of starch. C, separating colored compounds on a strip of paper. Or D, changing the genetic code of an organism. And the answer is A, we're separating DNA fragments. 3. In preparation of an electrophoresis procedure, enzymes are added to DNA in order to do what? What are we doing to the DNA? Are we A, converting the DNA into a gel? B, changing the color of the DNA? C, cutting the DNA into fragments? or produce longer sections of DNA? Your answer is C. We're going to cut the DNA into fragments using the restriction enzymes that we talked about in the beginning of the video to help break those fragments into those different sizes that are going to separate along the gel. Number 4, scientists hypothesized that cabbage, broccoli, cauliflower, and radishes developed along a common evolutionary pathway. Which observation would best support this hypothesis? A, these plants live in the same environment. B, these plants have similar proteins. C, chloroplasts of these plants produce a gas. Or D, fossils of these plants were found in the same rock layer. And we know that they all live in the same environment, yes, but we want to know why they are related through the evolutionary pathway, and we can figure that out by having similar proteins, and we can see those proteins and the DNA that codes for them using gel electrophoresis. So I'm going to go with B. Alright, so that ends our video. You're going to try 5 on your own. The diagram below shows the results of a technique used to analyze DNA. State one specific way that the laboratory technique could be used. Now just think of the different examples that I gave in the beginning of the video, and that is your answer for 5. Now you're going to continue on with what I've left you. I've left you another quick video to help explain DNA fingerprinting and how it's being used. You also have a homework assignment that is due tomorrow. Please don't forget that your other Schoology assignment is due as well. Have a good day, guys.